We are looking at our fifth PowerPoint here on the neuromuscular system. To be fair, we're spending most of our time just talking about the muscular system, but obviously your muscular system is under the control of your nervous system, which is why we're going neuromuscular in title here. So our objectives today, we want to understand how our central nervous system works when it comes to controlling our movement. And we'll talk a little bit about how the peripheral nervous system is doing that as well. We'll talk about the specifics of where your nervous system is interacting with your muscular system. So the junction where that's occurring, we call that the neuromuscular junction. And then we will talk about the makeup of skeletal muscles and what they actually look like and things like that. Then we'll talk about like what steps occur during the sliding filament model, which is actually what makes muscles contract. That's the sliding action of the proteins inside of our muscles. And we'll talk at the very end, we'll get into the different types of muscle fibers, which you guys might be a little bit familiar with because we talked about it yesterday. We were talking about type 1 versus type 2, but we're going to get even more specific about it. So that is our journey for today. So looking at muscles and how they are contracting, right? Muscles will contract because a nerve impulse, so a signal from your nervous system, creates what is called an excitation contraction coupling, which is like a very fancy way of saying a muscle contraction, but basically it is your nervous system exciting your muscular system. And so then the muscular system couples together and it causes it to actually move, right? See if I can find a GIF of this. Okay, that's a GIF, but it's not an animated one. Okay, let's do this one instead. Motor unit GIF. All right, so what you'll see is an impulse will run down and then it will spread throughout those muscle fibers, causing the contraction to occur, right? So you've got your spinal cord here, then you'll have a nerve that branches off that spinal cord, and then it will run down and it will cause the actual contraction to happen, right? So that's what we're seeing there, right? So if you have some type of like injury that interrupts your central nervous system, or an injury that interrupts your peripheral nervous system, that could interrupt these signals. This is what, like, if you think about like what paralyzation is, that is definitely something that's going to affect it. If you think about like any nervous system disorder, it will often affect like muscles. There's even like, there's things like Parkinson's, which is like involuntary contractions. That's a nervous system problem. If you look at like, oh, come on, brain. Oh, good Lord. I'm blanking out here. When they get really hot, they not cystic fibrosis and not cerebral palsy, the other one. Oh my gosh. It's an autoimmune disorder.
Muscular dystrophy.
Muscular dystrophy, thank you. Good Lord. If you look at muscular dystrophy, yeah, that's another one that involves an injury to the nervous system and prevents you, thank you so much, from being able to use your nervous system. And then multiple sclerosis, that's another one. MS is pretty fascinating, actually. MS is an autoimmune disorder as well, where basically the things that are protecting your nervous system, we call that myelin. It's just like a little fatty material that surrounds your neurons. Gets eaten away by your immune system. Like your immune system's like, I don't know what that is. That's a foreign invader. And so then it starts, eating at it. And then when that happens, like the electric signals are not protected. So then they might get interrupted. And so you'll see people like they'll lose their balance very suddenly. And then, they're able to catch it afterwards. Now, that is a really good example of like something that we don't necessarily have a direct impact on. Like we're working with a client and they're like, I have, MS, it's going to be very difficult for me to, exercise and stuff, but my doctor told me they want me to get like more physically active, right? So we can't necessarily like we don't have like a cure for this. Exercise is definitely not a cure for MS or other central or peripheral nervous system conditions, but it can have an incredibly positive impact on someone's overall like health and well-being. So let's say like we've got someone with like, MS, right? And they're going to lose that, like, every now and then their body loses the signal, and so their leg gives out on them mid-step. So if we have trained them to like develop their balance, develop their core strength, and their reactive capabilities, and they are able to like swing that leg around and get it back on the ground, that will keep them from falling all the way to the floor, which could prevent like a much more serious injury. So this is one of those things where like, exercise is not a cure, but it is something that leads to such a massive big improvement in overall like quality of life. We definitely it's definitely worth like studying and understanding. And I've had that conversation with so many different people before, where they're like, it's like diabetes as well. It's like exercise doesn't cure diabetes, but it does help you manage your blood sugar, which has a positive effect on your diabetes. And that is why, like, you will always hear me say, there's no magic, there's no silver bullet. Like I come from I'm gonna use this phrase a lot when we get to nutrition, because nutrition is the one where it comes up the most often. But I come from a long line of silver bullet people. And what I mean by that is like my parents love the idea of like a simple cure for something like snake oil that will just solve all their problems. Like my mom's got a back pain, so she gets one of those copper bracelet things. My dad found this like liquid vitamin that's like, oh, it's got all your nutrients in it for a day. My parents love that kind of stuff. I grew up around it. And the honest answer is a lot of times that there really is no silver bullet for anything. There's no, there's no, one step cure, but regular exercise and like, consistent healthy choices has a positive impact. And exercise is the one thing that really does impact very, very positively almost every single aspect of our lives. So, obviously I'm a big fan. So anyway, let's go ahead and take a look at some of the actual movement, right? So movement, very, very complex because there's a lot of stuff going on. So we're going to start by talking about the brain here just really quickly. Don't worry about having to have any of this memorized actually, but hopefully it gives you a little bit of perspective on stuff. So we'll come back to the notes in just a sec, but you do have different parts of your brain. You have what's called your precentral gyrus. It's located in the front of your sulcus, right? So if we look at the brain here, right, you can see these kind of different areas here, right? Each of these different parts of your brain do different things, right? You can see like here's where your speech patterns are. And then here is like your motor cortex that's really good for like, a lot of like complex movements. So like the front of your brain right there, right? And then there's other parts of it too. Your vision is back here. This is why the back of your brain, I think why. I'm definitely not exactly a brain expert. Where I'm definitely more on the muscle side of things. But like, if you ever fall on the back of your head and you smack the back of your head really, that's why a lot of times like it causes like blurry vision and nausea and things like that, because your visual cortex is basically right here in the back of the brain. So definitely not something we want to experience. Speaking of my parents again, my parents' neighbor George, he was, when he was a kid, he like was jumping on the bed with his brothers, and he like fell off and smacked the back of his head, and he's been blackout blind ever since. And for the record, he's my dad's mechanic. Like, and handyman. Which is amazing. It's really cool to watch him work. Like he's great at it. And it was like my parents' favorite thing. My cousin came down and my parents saw her car and they were like, oh my God, she's got kids and stuff. And it's like, we're gonna pay George to fix your car for you. Yeah. And she was like, okay, great. And then like later she met George and she like her eyes got really big. And she's like, this is the guy who put my brake pads on. Like, but he's a hell of a mechanic. So anyway, so that's, you know, the different parts of your brain, right? A lot of your movement is in the front of your brain and in the center of your brain, right? So you've got a little bit of difference here. You've got the front of your brain is used primarily for like involuntary movement and planning, and then the middle part of your brain is also like voluntary movement, and then this section right in here is this kind of dead center section is a lot of where all of your like sensory information comes from and involuntary movement. And that's one thing we got to talk plenty about. You know, a lot of times when we're thinking of our nervous system, we think of the stuff that we're controlling, right? We think of all the stuff that we are purposefully doing, but there is so much going on that is involuntary. You know, like your hearts are beating right now, your diaphragm's contracting involuntarily, even though it is something you have conscious control over as well. You know, these are, you know, certain areas where you're doing a lot of things that you don't want to have to necessarily think about, right? So that's the architecture of like your brain and sort of the neurology of movement there, at least in a very basic way. But if we're talking about any of this, all of this is going to rely on energy production, right? Which is why we have this exercise physiology course that we are currently in, right? This 12-day class where we just keep, you know, every day it's like we've been talking about, like, we talked about this part of the body, then we talked about this part of the body, but it all keeps coming back around all the time to like the idea of producing energy, right? And again, we can't really store pure energy, otherwise we would just, you know, but we can store ATP and we can generate ATP using nutrients and oxygen and things like that. So we have a lot of mitochondria, which are, you know, distributed within your muscle fibers, especially, but they're in your neurons as well, and they are there to produce energy for your cells, right? So we produce energy in two places within an animal cell, right? We produce energy within the fluid portion of our cell, and I'm just going to kind of pick a basic drawing here, right? We produce energy in this little fluid area right in here, but then little potato bugs, right? Those are your mitochondria. You know, this is where we produce lots and lots of energy. Like, again, going through it in a very basic fashion, right? For every 1 molecule of glucose, you will get 2 molecules of ATP in the fluid portion. But then you will also get a whole bunch of pyruvates, which will then pass into the mitochondria. And then with sufficient oxygen, those pyruvates will go through the Krebs cycle. That will give us 2 more molecules of ATP. So now 1 molecule of glucose and a little bit of oxygen gave us 4 ATP molecules. Then the Krebs cycle, once it's done creating the ATP, it also creates some byproducts in the form of like NAD and FAD, or NAD and FAD, as I like to say. And then that's going to pass into the electron transport chain, where we're going to get about 36 molecules of ATP. And that is how this cell is able to power itself and keep itself running. It's how its nucleus is able to create, you know, new DNA, and the ribosomes are able to assemble it. You know, that is how your cells, every type of your cell, is working. So your muscle cells have a lot of mitochondria because it takes a lot of energy to do all this stuff, right? So during a contraction, what's going to happen is you are going to have what's called a myosin cross bridge. We'll talk about myosin in just a second, but that's the little protein that's able to actually contract, where it basically breaks down an ATP molecule and converts it into ADP. Because remember how the way ATP works, right? Work? Yeah. The way ATP works, oh, look, it's a crash course picture. That is not animated. Oh, there it is. Right, you have ATP, so you have triphosphate, right? You have a adenosine and then 1, 2, 3 phosphates, and you kick that last one off. That's what breaks the bond, and whenever we break a chemical bond, energy is released. This chemical bond in particular has a lot of energy to be released, and so this is why this is our body's currency. So now this is ADP, ATP, And then we break the phosphate off and we're left with diphosphate. For the record, you can actually break the phosphate here as well and release some energy, but your body is not particularly good at it. And then you actually can even break down even a little bit further into AMP, adenosine monophosphate, but you don't have as much energy trapped in between those bonds. And AMP has actually been shown to inhibit muscle function in high levels because it basically just kind of fills up the area with something. It's like trying to run your entire house on AA batteries. It's like, we're going to need a lot of them. They're going to take up a lot of space and it's not going to work very well, you know? So that's how contractions are going to use ATP, right? So the primary function of ATP is creating enough energy to produce these muscular contractions. So finishing up the brain here, like I said, don't worry too much about this. You've got, your cerebrum, which is the really big part of your brain. That's like this whole like top section here. You can see like this area, this like whole area would be your cerebrum. Two major hemispheres, right? You get your left side and your right side, and they control like a lot of like that sensory function. So like when your eyes are letting in light and you're interpreting it and being like, that's the color blue, right? Like that's your cerebrum that's doing that. We've also got your cerebellum, which is basically kind of below that. And a lot of your neurons are actually located in the cerebellum, even though it's very small, because like this is eventually going to become, go into your brainstem. And so this is actually processing the information that you gathered in your cerebrum, which is kind of cool. Your medulla oblongata, traveling even further backwards. And that is what will eventually, travel all the way down the spine. So those are all different brain parts, kind of fun. I'm sure I'm butchering it a little bit because we're moving through that part quick. It's not super relevant for the way that we talk about the nervous system. We kind of look at it more on a global side of things. But then you are going to look at your peripheral nervous system. Okay, so when we look at our central nervous system, it divides, or when we look at our nervous system, it kind of divides down into different sections here. I don't know how to look up what I want to look up, so I'm just going to make my own. So you've got your central nervous, which is not how that's spelled, nervous system. Okay. You get your central nervous system, and it works together with your peripheral nervous system. Okay, so those are the two primary divisions of like how we are going to kind of divide things up. And they have like slightly different jobs, right? You can think of the central system as like the command center of your brain. This is where all the decisions are getting made. If you were to look at it in terms of like a factory, that let's say it's a factory that makes, some type of good, right? In the main office, right, there's the boss, there's the CEO, right? And he's in charge of like making all of the major decisions and which direction that company is going to go. That's your central nervous system. Okay. Your peripheral nervous system are all of the other workers that are working in the factory, who are actually carrying out the jobs, right? So if the central nervous system was like, what is this factory making? Let's say it's making kids' toys, right? It's like the central nervous system is going to go make kids' toys. And then he's going to tell the sales team is like, take those toys and put them on shelves in stores and charge this much money. So then the peripheral nervous system, which is all the workers, they're going to do that, right? So there's going to be the factory workers assembling the toys. There's going to be the salespeople who are going out and sales repping and getting toy stores to pick up their products and stuff. So that's your peripheral system, right? It carries out all the tasks. And then this is going to divide down a little bit into two other divisions, which is like the two parts of your nervous system that are either automatic or under voluntary control. Like I'm voluntarily choosing to like speak right now, but like again, my heart is not under my control, right? Like it just happens automatically. And so that's where we have what is called your somatic nervous system, which is voluntary, right? And then you have your autonomic nervous system. which is involuntary. That's the stuff that you don't have control over, right? So like all the little tiny contractions that are occurring without you having to think about it, those are all of your autonomic functions. And all the contractions that you're occurring that are, you know, doing bicep curls or something where you're consciously being like, I'm trying to work on my gun so I can get to the beach, right? That's you're using your somatic nervous system to control that. And then this does further divide down even further into two more divisions, which are going to be your sympathetic and your parasympathetic. You guys might remember we were talking a little bit about those sections yesterday. Your sympathetic system, that's good, drive me crazy. So, I don't know why I need to be straight, but I need to. Your sympathetic system is your fight or flight system. Right, and that's the fight or flight. When your body is freaking out about something, that's your sympathetic nervous system. Okay, that's you, like when you're panicking about something. You know, we were talking about panic attacks yesterday. That is your sympathetic nervous system functioning, right? And then we've got your parasympathetic nervous system, which is your rest and digest system. So that's the one that like calms you back down afterwards, right? And that's like a different section. So if you create adrenaline, that's happening in your sympathetic nervous system. It is autonomic. You didn't have any control over it. And your central nervous system made the decision, and your peripheral nervous system carried it out. Okay, so that's why we have this arrangement. You have the central system, which is like your brain and your spinal cord, and it's in charge of decision making. And then the peripheral nervous system carries it out. And that's what we're kind of seeing in this picture here, even though it doesn't look very detailed. But you can see, right? See these little numbers right here? It says C5, C6, C7, C8, and then T1. Those are actually your spinal discs. So if we look at like central nerve, you look at your central, actually, no, let's do central and peripheral nervous systems. If you take a look at this picture right here, you can see your brain and your spinal cord is your central system that is responsible for decisions. And then all of the nerves that branch out from there are what are actually carrying out the tasks determined by your central system. And let's see if I can get a picture with the vertebrae here. Hey, vertebrae can all be done on the left side of your keyboard. You don't need your right hand. That's kind of cool. It's a good one to remember for things like passwords. If you're a video gamer. All right, let's see here. No. Come on, give me. Let's see here. Vertebrae and peripheral nerves. Let's see if that gives me what I want. It's darn close. There it is. Okay, so if you take a look, you can actually see which parts of your body are sort of under the control of the different parts of your autonomic nervous system, right? So your C1, your C2 here, which would be called your axial vertebrae, actually. And that is the second from the very top of your vertebrae, right? So right here, this is your axis vertebrae right here. That's in control of, you know, all the sensations that you feel like in your neck and stuff. Those nerves branched out from here, right? They would basically be these little like tubes that would kind of branch out, and then they would spread and spread and spread until they're covering the area with nerves and stuff. You can see this section right in here, that's controlled by your C3. So go down a little bit further, and there's going to be, you know, further branches that are going to come out from that. And so that is what we're seeing in this version of the picture here. You can see all of these nerves that are branching out from the, oh, that did actually have, I just didn't see the numbers there. You can see all these little nerves that are kind of branching out like wires and stuff, right? So if you were to break your vertebrae and sever the connection from, you know, right here between your 4 and your 5, you would basically lose sensation in everything, most things like below it. Now your nervous system's pretty good at like drawing new connections actually, but obviously there is a certain sort of limit on all of that. But that's the different parts of your vertebrae in your skeletal system, actually, which is like housing these things that are protecting your nervous system where those nerves are branching out. It's a great picture right there. And then there's what's called your brachial plexus. It's basically the network. So that is the nervous system part of what we're talking about today. Any questions on any of that before we start diving into the muscle cell anatomy stuff? Conceptually, we're doing good. All right, everybody being quiet. All right, cool. So now we can get into the notes. So when we look at like the neurology of movement, right, your nervous system is going to control your muscular system through a series of what we call we call them action potentials. So that's one last thing I want to show you. Action potential. So we always say that your nervous system creates like electric signals, right? And that's because that's actually what it's doing. Your body is creating electrochemical signals. It does this thanks to its ability to store up electrolytes. So electrolytes, which if I look that up, it's immediately just going to show us a whole bunch of pictures of like powders and waters and Gatorades and things like that. But what electrolytes actually are is they are those chemicals on the periodic table that either have a positive or sometimes a slightly less positive or even negative charge, right? The big ones are going to be sodium and potassium. These are the two that we're going to spend most of our time talking about. But magnesium is an electrolyte that we are often lacking in. Chloride a little bit because you're going to get that associated with sodium. Calcium a little bit, right? But these are really the big ones, right? But the big ones are going to be sodium and potassium. So your cells have this really cool thing. Every cell you've got that creates an action potential, every cell that generates electricity, so nerve cells and muscle cells, especially the muscle cells in your heart, create action potentials, right? So if we look at what an action potential actually is, you will actually see where basically this electric signal, right? So he's like stepping, and then like it's causing this to kind of like travel down from point A to point B. And so you can actually see like, look at, oh yeah, it's a GIF, not a video. Try to pause it. Look at in this GIF, right? You see how like there's positive on the outside and negative on the inside? That is because when we look at the inside versus the outside of your cells, there is a whole bunch of sodium outside of your cells, which is very positive. And there's a whole bunch of potassium, that's the K here. Sodium is the Na, and potassium is the K. And there's a whole bunch of potassium on the inside of your cells. And so because of that, there's a different concentration. So these are actually both positive ions, but sodium is way more positive than potassium is. So we can think of the outside of your cell as polarized compared with the inside of your cell. They are opposites. When you think about that term like polar opposites, right? Or you get the north and south poles, right? They're opposite sides of the planet, right? That is like one's very positive, one's very negative. They are repelling each other away in the same way that if you flip two magnets around and then you try to like push them together, they'll repel each other, right? That is the electrons moving around, and they are pushing those forces away. So because you have a lot of sodium on one side and a lot of potassium on other, you have all this positive stored energy outside of your cells, which is why your cells have these really cool little pumps, right? That you can see here. There's that, remember phospholipid bilayer we're talking about, that fatty layer on the outside of all of our cells? That's separating this fluid on the outside, that's your extracellular fluid, and then it's separating all the fluid on the inside of your cells, that's your intracellular fluid. So because that fluid is separating, and then there's a suspension of all of this like sodium out here and potassium in here, they're very much kind of repelled in opposite directions. And what our body can do is it can actually have this little protein channel that straddles the membrane. And that's what it looks like right here, right? See how there's kind of different protein channels. There's the purple channel, the red one, and the green one here. They're all slightly different shape than one another. And what that allows is you've got like a sodium channel, which allows sodium to pass this way. And you got a potassium channel that allows potassium to move that way. And so you're actually, our cells, when they want to generate electricity and they want to do something, what they'll do is they'll flush sodium and potassium to opposite sides. And now you've got a little bit of positive stuff on the outside and a lot of positive stuff on the inside. And you got a little bit of negative stuff, right? So you flipped it, right? It's like taking a battery and flipping it around. All the positive became negative, all the negative became positive. And that's what we're seeing. And then this action potential is going to travel down our neurons. So your nerve cells are covered in these pumps that are pushing sodium and potassium in opposite directions. The example I always show people is it's literally, hold on just a second. I literally keep a dead remote in my little drawer here just for this example. I don't know why I don't just keep the batteries. Wait, that's not it. There it is. Oh my God. Did I steal my own batteries and screw myself in the future? Oh, past Brad, that guy's always making problems for me. God damn it. All right, well, we'll use this one for my current house. Well, all right, I guess I can. So take a look right here. We have one battery. I don't know where my friggin' second one is. So what's happening is like we are storing a bunch of energy. Now, if I were to put this battery with another one inside of like a TV remote, and let's say I put them both facing this way with the positive side up and the negative side down, what would happen with that remote? Nothing, right? Like nothing would happen because electrons are not moving between the battery. But if I took the right battery and if I took the left battery and I left it upright, and then I took the right battery and I flipped it around like this, now I've got the positive and the negative sides, I've got them flipped. So what's going to happen is the electrons are going to try to naturally move to where they can get into a position where they're all reunited. But what that does is now the electrons are moving like crazy, and my TV remote would capture those electrons as they're moving and use it to send an electric signal to my TV. And that's exactly what's happening in a remote. That is exactly what's happening in your cells. So your cells are moving sodium and potassium, and it's changing the concentrations to where it is flipping them, and they are polarizing. And then what will happen is they will depolarize and get back to normal afterwards, because nature does not like it when it does this. It likes to get them nice and even back on both sides. Remember, nature hates gradients, right? This creates a gradient where there's a lot of stuff on one side and very little stuff on the other. So it naturally wants to move the other way. So if it naturally wants to move the other way, how the heck do we resist that? We burn up ATP. So ATP is what causes those channels to move and shoves the sodium and the potassium in their different directions. This is also one of the reasons why, like, if you're ever cramping up, you know, they'll say, oh, go eat a banana, right? We say go eat a banana because most people are potassium deficient, because like we live in America, we're getting plenty of sodium for the most part. But if you have a really healthy diet and you start cramping up and you're like, I don't get it, I had like a banana smoothie this morning, I don't know why I'm cramping up, you could actually be lacking sodium. I know that sounds crazy in a world where we have salt on everything in the US, but it's true. Like, you could actually be lacking sodium. So this is how we generate action potentials. We flush the concentrations of sodium and potassium. That creates an electrochemical gradient, which nature hates, because nature hates gradients. So then it will naturally restore the concentrations back to normal without spending any energy, for the most part. And then that we are able to generate that energy again. And then that's how we are sending signal after signal after signal, by burning up a little bit of ATP to move those channels. How's everybody doing on that? Does that make sense to everyone? I want to get a good verbal confirmation from everybody.
Yes, sir.
Cool, cool. Paula, how you doing? Thomas, Bay?
This does.
Maybe. All right. I'm seeing a thumbs up or two. Okay. Cool. So that's how action potentials work. If you have questions, please let me know that we can make it, much more clear. This, by the way, here you go. You can see again, right? Channel opens up. That is going to be, the concentration's moving, right? So all of that sodium comes rushing in, right? And that causes, the action potential, which you can see, like, here's the amount of electricity, right? So once it hits this number right here, 40 millivolts, that is actually what triggers our contractions. Now, there are certain things that sometimes cause this to happen, where like you might sense something, for instance, like if something really gentle touches you, but if it doesn't create a large enough signal to get 40 millivolts worth of electricity, the signal doesn't take. It's sort of like a false firing, and then it just goes back to normal and repolarizes, which is kind of cool, actually. But that's what we're seeing in I don't know how to look it up. Action potential. Let's try that. There we go. This is what we're seeing sometimes when you look at like the action potential of things, right? Now, what's kind of funny is when your body restores the concentrations back to normal, it actually does a little bit too good of a job. And so you'll actually see this little dip right here that goes below our actual resting potential before that concentration gets back to normal. So this actually should look pretty darn familiar to some of you guys. If you've ever watched any medical drama, Lorianne, I know this looks incredibly familiar for you, because you've actually, been around these monitors for a while. But this pretty darn similar to the action potential. This is what we're looking at if you ever see like what's called an ECG. Okay, good. Notice how like you get a big spike and then it actually drops down below, right? You're actually seeing the action potentials, that your heart is creating. This is how heart rate monitors work. If you've ever worn like at least it's how the chest strap heart rate monitors work. If you've ever worn one of those, it is actually picking up every time 40 millivolts gets created in your heart. It knows when it happens. It's the sensor is picking up on it. And so then it's going, okay, well, we created 10 of them in that 6 seconds. Your heart rate is currently 60 beats per minute. Kind of cool, right? And that's why the chest strap monitors are super, super, super accurate. Now, I don't hate these type of monitors. If you wear the wrist ones, I mean, like, who wants to wear a freaking chest strap all day? But like, if you're wearing one of these style with like the green light behind it, that's also going to tell you your heart rate, but it's actually just counting every time it watches red blood cells like rush by, which the faster your heart rate goes, the more it starts to look like one big fast flow. And so these get a little bit less accurate the higher your heart rate goes, but they're pretty accurate at a low resting heart rate. And then the chest strap ones are accurate all the time. So if you're taking your cardio seriously, if I've got anybody out there who's looking at getting into marathoning and stuff, get yourself a chest strap heart rate monitor. They are the best. All right, so that's an action potential. That is how we are using sodium and potassium to create electrochemical signals. That's how one neuron is going to communicate with the next neuron, right? When that neuron zaps that neuron will zap that neuron, which will zap that neuron, and that one, and then that one. And eventually it'll get all the way down to your calf muscle, and you'll walk forward, right? So that is how the nerves are communicating. They are zapping each other, right? Just like if you picture, like, if you've ever seen that video where like a group of people grab electric fence. So here's an electric fence, right? And this group of people are going to grab it. And this last guy is the one who, you know, kind of causes the whole things to happen. So everybody's insulated currently until this last guy who is not insulated is going to grab. And so electricity is currently running through all of them, but it's grounded. So it just kind of goes back into the wire. But now this guy's going to grab them and they all get zapped because he wasn't grounded at the end there. But like, you know, electricity is running through each person right here. That's exactly, you know, neuron to neuron to neuron to neuron to moron. I mean, obviously this was all on purpose, but for the record, the same thing happens in your muscles as well. You do have action potentials in your nervous system. That's usually where we spend most of our time talking about action potentials, but you do also have action potentials that run across the surface of your muscles as well. So your muscles will spread an action potential along them, and that's how all of the individual fibers in your muscles contract. So that's enough about action potentials. Let's move on and start talking about the muscle fibers themselves. Okay, so inside of a muscle, let's take a look at some muscle anatomy here, right? So when we look at like an animal cell, for the record, none of your cells actually look like this. I know I pull this picture up all the time, but none of your cells. This is just kind of a textbook in an animal cell, because like all of your cells have these pieces in them, but they very rarely look like this. The closest you can get actually to this type of cell would be like a fat cell. If you look at an adipocyte, which is a fat cell, it actually kind of looks similar, right? Because it's got like the normal cell stuff right in here. And then there's this little extra sac that it has inside of it where it stores triglycerides. And that's the fat part of the fat cell. So no cell actually looks like this. But every cell has all these pieces. It has mitochondria and endoplasmic reticulum. It has like a nucleus. So muscle cells look like this. We call them muscle fibers. Okay, so muscle fibers, which is a fancy name for a muscle cell, look a little bit specialized. It has all that normal stuff and more. Muscle fibers have these special materials in them called myofilaments. Okay, so when you look at a muscle cell and it's got, I guess this will have to do because I'm not getting a better version. Trying to find a better version. Yeah, no, I'm not gonna find one. Because it would actually be kind of a poor way to teach it. Actually, you know what? That's not, no. Nope. Yep. I'm going to be here all day if I try. Okay. So here's kind of an individual, like, you know, muscle cell, right? And you can see like this little like tubing, this like webbing here. We call that your sarcoplasmic reticulum, which is like a specialized version of the endoplasmic reticulum, which is this stuff right here, right? So this like little like webbing, the endoplasmic reticulum, it gets a special name in muscles. We call it the sarcoplasmic reticulum because the prefix sarco means flesh in Latin, I think. And so anytime you see the word myo, it means muscle. Anytime you hear the word sarco, it means flesh. So a lot of our anatomy is going to either have the prefix myo or sarco whenever it is we are talking about muscles, okay? So again, it's a normal animal cell. It's got all the mitochondria, it's got nuclei, it's got ribosomes and cellular plasma, which we call cytoplasm. It's got all the stuff inside of it that every cell has. It just looks wildly different because it needs to. It needs to look different because it has a different function, right? Your muscle cells have to do a lot of contracting, and so they look very different, right? They're kind of stretched out like this. So this is kind of what you'll see in a normal muscle cell, right? You can see there's a nucleus right there. Can't really see any mitochondria, but trust me, there are a ton of them running throughout this muscle. What we are paying attention to, what we see a lot of, are these little tubes right here, right? We see all these individual little tubes here. Those are what we call myofibrils, and that's really what we're spending our time talking about today. So inside of a muscle cell, you're going to have all your normal organelles. And you are going to have what are called myofilaments. myofilaments, okay? Myofilaments are special proteins that are able to bind to each other, and that actually allows the muscle cell to shorten. So muscle cells are able to go from being this long to being this long, and then they can relax back to their normal length. That is contraction. This is why your muscles kind of bunch up a little bit, actually. Actually, I gotta lose my sweatshirt to be able to show you this. So if we look at like how muscle cells are kind of bunching up, they're sliding across each other. So you can even see, right? If I go from my muscle being really long like this, it's all stretched out. Everything is nice and relaxed. But if I bunch it up like that, right, you can see it kind of creates this like little peak, right? That bunching action is the sliding. So you're stacking everything together. So it all kind of bunches up. Those are the myofilaments sliding across each other, okay? So myofibrils would be where it's stretched out end to end. So a myofibril would be like the entire thing. The myofilaments are the individual proteins that we're talking about. And those individual proteins are going to be actin and myosin. So if we look at like actin and myosin, which you can see here, right? Like look at this picture. You can see this big tube and then see all the like little crossing. There's like the little blue lines and there's little red lines. Well, let's zoom in like crazy, right? Let's zoom in a lot. Well, what those actually look like is this. You can see right there's the crossing, and then we zoom in and it looks like that. And we zoom in and it looks like that. This is where contractions actually occur. Wrong picture. So you've got this little blue strand here. That's the thin filament. And then you got this really thick filament in the middle that is got these little heads on it, right? You see the little like tiny circles that are on there. Yeah, there's an even better. That's a much better picture. Right, you can see this like thick band in the middle, and you see these little thin filaments. So this is actin, which is the thin strand, and myosin, which is the thick strand, okay? This is what allows contractions to take place, like I said. So inside of a muscle, you've got actin. It's a thin filament that has all these little like binding sites on it. It's got all these little spots for myosin to reach up and kind of dock with it, right? And then myosin is the thick filament that has these little heads, these little club head things. They kind of look like golf clubs. And they reach up and grab, and that's what allows it to pull. So if I've got like a binding site here in this hand, and then I reach up and I bind with it, and I pull it this way, right? I'm sliding my actin this way. Now, that causes my muscle to shorten on this side. And if I got the exact same thing happening on the other side, it goes like this. And my muscle cell goes from being this long to being this long. And then it can relax back to normal. So actin is the thick strand. Nope, actin is the thin strand with all the binding sites. That's what we're seeing right here. And myosin is the thick strand that has all the heads that bind to the binding sites. Now let's take a look at that in a GIF here, right? Let's take a look at like what that actually kind of looks like. Actually, for the record, it kind of looks like this. Kind of just drags it along with it, right? Like, I love these type of cell pictures because it looks so funny to me. They look like Muppets. But here's kind of a better version, right? Here's kind of a crawling action. And you can see the muscle is shortening, sliding across each other, right? That is the sliding of the sliding filament theory or sliding filament model that those two scientists both discovered on the same day. They both determined that there are proteins that are sliding that are causing our muscles to contract. Now, obviously, like I said, myosin, its little club-like heads, they are super attracted to actin. They really want to reach up there and bind. So we don't always want our muscles to bind. We want our muscles to be able to contract sometimes, right? So because of that, we have these two regulatory proteins, which are called troponin and tropomyosin. And troponin and tropomyosin, you can actually see it right here in this picture. Ooh, please be big. Yeah. See this little like band that kind of like lives on this, right? This little strand right there. That is your tropomyosin. Okay. And then you see this little like blue dot that's attached to the band. That's your troponin. So troponin and tropomyosin live to block these binding sites, but when they're dragged out of the way, that allows the binding sites to become exposed. And that's where the heads reach up and grab. So that's why we're able to relax our muscles, because these chaperones, right, these little, like, little binding site blockers get in the way. And so myosin can't get in there, right? He's being blocked. And so he can't reach up and grab until we move those chaperones out of the way. If we can move troponin and tropomyosin out of the way, now sliding can occur, right? So how the heck do you get troponin and tropomyosin to move away? How do you move those out of the way? Well, you actually use a little bit of calcium, which seems kind of weird, but you didn't know that calcium was going to be a big part of the conversation when it comes to muscle contractions, but it absolutely is. Troponin and tropomyosin are very attracted to calcium in the same way that myosin is attracted to actin. So when you flush, if you were to take liquid calcium and just inject it into a muscle, it would actually cause your muscle to contract, because those binding sites would become exposed and your muscle would contract. So you can see this is calcium right here getting into the area. That's what drags the binding site blockers out of the way. Now, you may be saying, okay, so I know that actin wants to bind to myosin, or well, no, the myosin wants to bind to actin. Got it. And I know that it can't do it unless the binding sites are out of the way. Got it. And then you might go, okay, how do I get the binding sites out of the way? That's right. I need calcium. Calcium exposes the binding sites, right? How the heck do we get calcium into the muscle? Well, that's where the action potential comes in. So your muscles will store a bunch of calcium ions in the sarcolemma, right? They'll store it in the muscle cell area, right? It will store all that calcium. And when that action potential from your nervous system zaps a muscle cell, that opens up the gates and flushes calcium into the muscle. When you flush that calcium in, that will cause the contraction to occur, because you are moving the chaperones, which exposes the binding sites. So our nervous system uses sodium and potassium to generate electricity. And it zaps one neuron, then the next neuron, then the next neuron, then the next neuron, until eventually it gets to a muscle. Once it reaches a muscle, the neuron zaps the muscle. The muscle then creates its own action potential that runs across it. And as that electricity runs across the muscle, that causes all the stored calcium to be released into the area. When calcium moves into the area, troponin and tropomyosin move out of the way. The binding sites are exposed, and actin and myosin are like, it is on, and they get all up in each other's business and create a contraction. And that whole process that we just talked about is the sliding filament model, or the sliding filament theory. I don't know what they call it anymore. It was theory when I learned it, and now I keep hearing the term model all the time. So sliding filament model. And so basically, and all of this takes energy, right? So an ATP molecule is broken, that releases the energy, which triggers the action potential, which allows the sliding filament model to take place. Whew. It's a lot. It's a lot of working like a lot of working pieces. But they want you to know this kind of stuff, because it is going to play a direct role in how we train our clients. It depends on like what type of strength you are trying to train your clients for, right? So let's say Paula wants to, work on improving her 1 rep max on a barbell squat, right? She's trying to like get a new, she's trying to like squat 300 pounds, right? Not making any assumptions. Maybe you can already squat 300 pounds, but like maybe that's your goal, right? And then Lorianne wants to, have more sustained endurance, because maybe she, wants to run like a 5K, right? So now we've got 2 very, very, very different goals, right? That are, both involving training my muscles, One involves a little bit more cardio, but like both involve like using muscles, but using them in very different ways. Paula is going to use it for a very short period of time for a lot of intensity. And Lorianne's going to use it for a very long period of time for very minimal intensity per contraction. So if I understand the anatomy behind the sliding filament model, I know that I'm going to have different energy needs for both of these people as I'm training them. Paula, I want to train her to be able to use a big burst of energy. And I want to give her plenty of rest in between rounds to make sure that she has all of the energy she needs. And I'm going to spend a lot of time training her nervous system, how to create action potentials as fast as possible, how to make sure that they're synced up, and how to make sure that they are recruiting lots of them. So how do I recruit lots of them? I'm going to use compound movements, big, heavy movements that involve a lot of muscle. How do I train to get them to make like a big burst of energy? I'm going to keep the repetition duration pretty short. And how do I make sure that they're all synced? I'm going to give her lots of rest between sets. So now suddenly it's like, oh, okay, well, why don't you have them do 3 repetitions for a lot of weight in a big compound movement and then give her 3 to 5 minutes of rest? And guess what? That's exactly what it looks like in your textbook. 1 to 5 reps, 85 to 100% of your 1 rep max, 3 to 5 minutes of rest. For those of you, and this is just going to be Lorianne, Bay, and Thomas here, those are some acute variables from our program design class. Our understanding of the sliding filament theory has led to creating our sets and our reps and our things like that, right? Is directly tied to what we're doing. Lorianne, very different goal, right? Her goal is to develop like endurance, right? She wants to run a 5K. Great. If that's the case, I need her to be able to create energy consistently. So I'm going to give her very little rest. I'm going to give her very minimal weight. And I'm probably going to have her do lots of stabilization so that we can strengthen like the stabilizing muscles as she's running through that whole race. So I'm going to pick an unstable exercise type, something that involves like a little bit of balance. I'm going to have her do probably 12 to 20 reps at the very lowest. And I'm going to have her lift at like 50 to 70% of her 1 rep max. And I'm probably only going to give her 0 to up to 90 seconds of rest at the most, but 90 is a lot. I'll probably only give her 60 seconds of rest. So sometimes, I want this to kind of serve as like a reminder here. Sometimes it seems like we're talking a lot about like this anatomy and this physiology, and we kind of lose sight of the fact it's like, how does this apply to the gym? Well, it's what created our understanding of what to do in the gym, you know? So that is the sliding filament theory. Now, in your nervous system, you do have what are called motor neurons, or in this case, we're calling it a motor nerve. These are the types of neurons in your nervous system that interact with your muscles. So you got a lot of different types of neurons out there. You got sensory neurons. They interact with like your senses, right? You got chemoreceptors, they resense to chemical changes, right? Well, motor neurons are the ones that are, you know, connected to your muscles. So a motor neuron controls your muscles, okay? So when we see like a neuron travel down, the neurons we've been talking about today are basically all motor neurons. And what happens is an electric signal will travel down a motor neuron, and then it will zap the muscle, creating excitation contraction coupling. It describes where the motor neurons are innervating, zapping your muscle cells, right? So it would travel down and, you know, create all. I thought I was going to get a picture there. Am I going to get a picture? Yeah. So here's where that's actually happening. Oh, by the way, you'll also see there's different layers to your muscles. I didn't put that on here, did I? Yeah, good. There are different layers in your muscles, your muscle cells. Don't worry too much about that for now. This isn't going to come up in this class, but yeah, you do have like the outer layer, which is like your fascia. And then each layer, each bundle of a bundle also has its own like little connective tissue that's surrounding it, different little bits of fascia and stuff. So here's our motor neuron, right? You can see we are very zoomed in here. This is the very tail end of a motor neuron. Let's take a look at a motor unit so we can zoom out a little bit. So, we're basically just zoomed in right here on this little, connection point. So here's a neuron, there's its tail, and there's where it's interacting with 1, 2, 3, 4 different muscle fibers, right? Or 4 different muscle cells. So that's a motor unit. So a motor neuron is a neuron that interacts with a cell. And that cell might interact with that neuron might interact with 50 to 100 or maybe even a couple thousand cells. That's what a motor unit is, right? So a motor unit is several muscle fibers. They're all working together under the control of a single neuron, okay? You've got small motor units in like your eye, for instance, right? Moving your eye around, like probably only a couple little muscle fibers there. And you got huge motor units in your glutes, right? Because that's the most powerful muscle in your body. So those motor neurons are going to create excitation contraction coupling with several muscle cells. We would call that a motor unit. And then I put this in here. I don't know why this is down here, but a muscle fiber, we've used this term already. It's a muscle cell, okay? So what the last little bit here is what is actually, so we said like the action potential from a neuron creates the action potential in a muscle. That is true, but it's not just because the electricity just straight up passes from one place to another. We have to tell the muscle to create its own action potential. We have to tell the muscle to do the sodium potassium swapping, right? So how does that happen? Well, yeah, like I said, it's not just electricity that occurs. What happens at the end of your neuron? Take a look right here. See these little pockets, and they have these these little dots on them. So there's a little gap between where your neuron is connecting with your muscle cell. That gap is called the synaptic cleft. It's this little area where, if it's got a little, dent in it like this, there's just a little gap, a little bit of space in between the two. And what's going to happen is when the electric signal, the action potential, gets to the very end of the neuron, it will release these little red dots here, which are neurotransmitters. Neurotransmitters are chemicals, and that's how your nervous system actually controls all the stuff inside of your body. It releases neurotransmitters. The neurotransmitter we're talking about here is called acetylcholine, or ACH. And so acetylcholine will move across the gap and bind to the muscle cell. Once it binds to the muscle cell, that's what triggers the action potential in the muscle cell. So now the muscle starts generating an electric signal, flopping sodium and potassium, right? That's what, again, triggers the release of the calcium, right? The calcium flushes into the area, and that causes the contraction to occur. So if we look at this, that synaptic cleft, the neuromuscular junction. Here we can see the action potential traveling down, right? And then what's going to happen is once it reaches the end, oh, actually, sorry, it didn't quite show me what I wanted. Oh, internet, you are so reliable and yet still so unreliable sometimes. That's the same one we just looked at. I'll take a simple one. Yeah, there we go. That's perfect. So what happens is the electricity, the yellow bit, travels down, and then that releases all this neurotransmitter. When that happens, the only way I would improve this GIF is then the electric signal is going to run across the entire muscle. So that trigger, that's how your nervous system's communicating with your muscular system, right? So we'd see the electricity split. This is a motor unit, right? Fiber here, several different fibers. And then that is going to release the acetylcholine into all of this individual muscle fibers. When that happens, acetylcholine is what triggers the release of calcium, okay? So one more time, let's go through every step in detail, right? And then we'll move out of this section. So what will happen during a muscle contraction is acetylcholine will degrade and move across from the neuron over to the muscle cell. As that happens, as it binds to little sites that are on the outside of the muscle cell, that triggers the muscle cell to create an action potential. That action potential runs across the muscle, triggering the release of calcium into the sarcomere of every one of your individual like muscle fibers. Once that calcium is released, troponin and tropomyosin move out of the way because they are attracted to the calcium. That exposes the binding sites on actin. So then myosin's like, let's do this. And so it reaches up and grabs that binding site and then drags it. And that's going to happen in this muscle part, half of the muscle cell, and that's going to happen in this half of the muscle cell. So my myofibril is going to slide like this and shorten, and my muscle cell is going to get back to here. And that takes a lot of ATP and split little energy molecules, right? Now, how do we get it back into normal position? Well, ATP will then actually, the burnt up ATP will detach, and then a new ATP molecule will come in and attach. When that attachment takes place, that flushes the calcium out. The chaperones go back into place, and myosin lets go and goes back into a resting position, where once it gets back to its resting position, it'll break that ATP molecule and get ready for the next contraction. And that's the whole process, right? That's the whole process of the sliding filament model. And again, two different groups of scientists discovered that on the same day. And they both had the same last name, and they were not related to one another. What? All right. Anyway, I did a whole here. The reason I'm making a big deal out of this, I'm going to fill you guys in on some backstory here. We had to choose to write a paper. It was supposed to be a research paper. It was supposed to be like a science paper in school. And I had to write like a thing. And I wrote it, and I spent so much time focusing on how crazy that story was because it blew me away. And I got like a C- on that paper, because like they were like, we asked you for a science paper, not a science history. And I was like, come on, that's the coolest story. And they're like, yeah, we don't care. We just want to know that you understand how contractions work. And I was like, yeah. So now that I'm a teacher, I'm getting my revenge on the world. I'm justifying it. Okay, so let's get into the easier part of this lesson. Let's move out of the chemistry. Let's move out of the little proteins there. Let's take a look at how we actually like move some stuff around, right? So when it comes to creating contractions, you got three different types of contractions. I know that we think of like this as contracting, right? We think of like shortening your muscle as contracting, right? But actually lengthening your muscle is also a contraction. It's just a different type of contraction. And if you're holding your muscle in one spot, that's also a different type of contraction, right? So we got three types of contractions. You have got the one that's probably the most famous, which is going to be a concentric contraction, right? This occurs when your muscle is shortening. So if your muscle is generating, let's say, I don't know how much this weighs. This probably weighs 3 pounds, right? If I generate 3 pounds of force in my deltoid muscle here, right, and 3 pounds of force in my tricep, right, I will overcome this little 3-pound weight here. That's a concentric contraction. My muscle fibers are shortening. They are sliding, right? So when your muscle is developing greater tension than a resistance, you will create a concentric contraction. The opposite of that is, what about like relaxing your muscle down? That's going to be an eccentric muscle contraction. So an eccentric contraction occurs when your muscle is lengthening. When your muscle is developing less tension than whatever resistive force you've got, that's an eccentric. When you're setting the weight back down, right? That's eccentric. And then isometric is a term that describes like constant tension. So if I had 3 pounds and I generate exactly 3 pounds of force, I can just hold this here, right? And I can just hold it kind of passively. If I create more than 3 pounds of force, I'll contract and shorten. If I create less than 3 pounds of force, I'll lengthen. So concentric, eccentric, and then if I just hold it, that's isometric, right? Those are the 3 types of contractions you've got. Now, we will label this out when we are describing it to our clients. Paula, it's the first time you get to see our workout template here. So here's our little OPT template, the way that NASM likes to write out workouts. You can see, we got our warm-up section, our movement prep section, any speed ladder work and things that we're doing, and then resistance. We'll just kind of jump to resistance here. Let's say we're going to do total body, right? So we're going to do like a shoulder press. We'll do a chest press. We'll do a bent over row. Then we'll do squats and calf press. Very simple, right? Kind of work our way from top to bottom on the body. So let's say I was just doing 3 sets of all these, and I did 10 reps for each of these. This isn't meant to be the world's greatest workout or anything. It's just very basic. Now, under tempo, I got 2 ways I can write this. We want to teach you the very technical way, and then the way that your clients are going to understand it. If I want to say slow tempo, I can. Whoops. Well, I thought I could, but apparently I can't type. So there's slow tempo. So I can just say that for my client, right? And then let's say I give them, 60 seconds of rest between sets. Okay, so my client, if I mean a slow tempo, I'm probably just going to describe to them, in your shoulder press, I want it to look something kind of like this, right? You're going to push, and then you're going to hold for just a second, and then you're just going to bring it down nice and slow. Push, hold for a second, bring it down nice and slow. But we want to be a little bit more technical about how we describe this. What if I really want to describe how long I want each part of the contraction? Because there's 3 parts to every rep, right? There's the concentric part, the eccentric part, and the isometric part, which is in the middle. So if I wrote my tempo like this, 4, 2, 1, this is what we call a stabilizing tempo. This is the type of tempo that we use to train someone's nervous system to learn how to do the movement. If I'm doing my shoulder presses like this, my nervous system doesn't have very much exposure to the exercise. Like 1, 2, 3, 4. I'm done with 10 reps in a couple seconds. But if I move really slowly, my brain learns how to there'll be a second where maybe I tilt a little bit too much, right? And I fall out of alignment. And then my body was like, whoop. And now it learned, I gave it time and exposure to learn how to get back to neutral position, to get back to good form. So we use slow tempos to indicate to our clients that we want them to learn how to do the movements. Now, do I write 4, 2, 1 for all my clients? Not really. I just write slow. But I know that slow translates to 4, 2, 1. Now, when I say 4, 2, 1, what the heck do I mean? Well, the first number, whenever you're writing tempo, bigger, even bigger. Yeah, there we go. So when I'm writing out tempo, right, and I've got 4, 2, 1 right here, right? This number here, this first number, always, always, always represents the eccentric contraction. Okay, this will always represent, oh, you mother. God dang it. The eccentric part of the contraction, okay? That's the number that we always put first, okay? The lengthening part of the contraction. And then this number right here, the middle number, that is going to represent my isometric portion of the contraction, okay? So this is isometric. And then this part right here, the third number, is going to represent the concentric portion of the contraction. Okay, we always, always, always put it in this order. This does not represent, let's look at a shoulder press, right? Let's say I grabbed a dumbbell, I'm holding it here, and I'm going to start counting reps. I'm going to start counting from here. The first part of the movement is concentric. So does that mean that I put the 1 over here? No. When I'm labeling it out, the first number is always the eccentric portion of the lift, even if your lift starts concentrically. I feel like the reason we did this is because trainers' favorite exercise is squats. And whenever you're squatting, the first part is eccentric, right? So a 4-2-1 squat tempo would look something like this, right? I would go 1, 2, 3, 4, 1, 2, 1. 1, 2, 3, 4, 1, 2, 1. That is how I'm going to learn how to do a good squat. My brain is going to be exposed to the lift for a substantial period of time, which is going to give me time to memorize it. I could write this out as a 2-0-2 tempo if I wanted to, right? I could very quickly just change that tempo to 2-0-2. And it would be the exact same thing. I'd still be labeling it out as 4-2-1, right? Eccentric first, isometric in the middle, concentric at the end. So that would be 1, 2, 1, 2, 1, 2. It's a little bit fast. 1, 2, a little bit slower, right? Just like that. Bicep curls, right? 1, 2, 1, 2. 4-2-1. 1, 1, 2, 1, 2, 3, 4. 1, 1, 2, 1, 2, 3, 4. Tricep extensions. 1, 1, 2, 1, 2, 3, 4. Right? They're all labeled out that way. Okay. So those are your different types of contractions, right? Concentric, you generate a greater amount of force than you are using as a resistance, right? Eccentric, use a less amount of force. Your muscle is generating less force than a resistance. And isometric, your muscle is creating an equal amount of force. So isometric and eccentric contractions are heavily associated with teaching your nervous system how to get into specific positions. And that's why we are stressing them when we are doing stabilization lifts versus like maybe I'm trying to develop like muscular growth. Well, if I'm trying to grow my muscles and make them larger, well, I'll probably focus more on a 2-0-2 tempo. And then what if I was trying to go really, actually, let's put this over here. What if I was trying to teach my muscles how to get real powerful, real strong? Well, then I might use an XXX tempo. And XXX basically just represents an explosive tempo that is used to like, you know, create like a lot of like muscular strength and muscular power. So we can think of this versus this versus this. We can think of it as stabilization. We can think of this one as strength, hypertrophy. That's a fancy way of saying muscle growth. And we think of this one as max strength / power. I made these all way too big. So those are our stabilizing contractions for clients who are learning how to do a movement, beginner clients. These are really good for hypertrophic growth, and these are really good for maximum strength or power. Okay, and those are the different tempos. I know that sounds really familiar to a lot of you guys. Lorianne, Bay, Thomas, you guys have already seen these when we get through our program design classes. But Paula, these are some of the things you'll have to memorize when you're writing programs for your clients. You have to consider like, what's their goal? You know, what do they want to work on? And then you're going to match which tempo, which rep range, which everything kind of goes with it. And so that's why we have this physiology class, right? You guys are getting the why behind the OPT model, the why behind the reps. Any questions on that? Cool.
Can you describe the hypertrophy that
Oh, hypertrophy. Yeah, it's the worst. That word is terrible. Hypertrophy is muscle growth. The reason why 202 works really well for muscular growth in particular is because notice how you're not taking a pause in the middle. So it's pretty much constant movement. So constant movement is going to generate the most lactic acid because it's not going to give lactic acid like any time to like get out of the muscle. So you're just like, by the time you're done with one rep, you immediately create the other. I always like to think of it as like friction, right? If I went like this, my hand has a chance to kind of cool down for just a second in between each like thing. But if I go like this and I just have constant movement, my hands get hotter and hotter and hotter and hotter. So I'm creating more damage in my hands. Eventually my skin's going to start to flake off, right? Well, we are trying to create maximal cellular damage when we're in hypertrophy, because maximal cellular damage will translate to maximal muscular growth in terms of size. So hypertrophy, which means, a trophy means growth, hyper means lots of. So we see lots of growth. It means a growth in muscle size. Whereas like atrophy means, a shrinking of muscle size. Does that make sense?
Yes, thank you.
Yeah, of course. For the record, you will also get muscle size from this one here if you're going for strength and power. It's just that NASM believes this is kind of the best version. There's a little bit of debate on that one in the scientific world, but because there's kind of two different types of hypertrophy. But for our purposes, that's a pretty good understanding of it. All right, so last thing we have to look at is how our muscles grow in different various ways. Let's take a look at our different muscle fiber types. So we are going to divide muscles. And by the way, all of your muscles have the same components in them. It's just the distribution of what those components look like might be a little bit different if you have more mitochondria or less mitochondria, if maybe there's more blood vessels running through the muscle, maybe there's less blood vessels moving through the muscle. So there's a slight difference between the muscle fiber types. And so we're going to divide them into two major categories. You're going to have type 1 fibers and you're going to have type 2 fibers, okay? Type 1 fibers are what we refer to as slow twitch oxidative fibers. Slow twitch meaning that they don't create a lot of forceful contractions. They contract, but they're not used for like maximal lifting. And then oxidative fibers, meaning they get lots of oxygen delivery. So they're really good at just working consistently because they got lots of mitochondria to be able to, just spit out energy all the time. So these are muscle fibers that have a very well-developed respiratory system. And again, we're not talking about the lungs in this case, we're talking about the cellular respiration, which ensures the delivery of oxygen to the muscle fiber. So when you look at a slow twitch fiber, you will notice it will be a little bit smaller in size. It will have a lot of mitochondria in it. It will have greater hemoglobin levels in your blood cells, and then you will have greater myoglobin levels within the muscle. Remember, hemoglobin is found in our blood cells. Myoglobin is found within the muscle. Hemoglobin drops off oxygen, and then myoglobin carries it around the muscle cell wherever it needs to go. And so these type of fibers are used for long-term endurance type contractions. It's always easy to tell when you look at the difference between a sprinter versus a marathoner. When you look at the difference between these two athletes, right? Very, very different looking, right? You can see a sprinter's body here. It's very muscular in terms of like hypertrophy. These are large muscles versus a marathoner. These are very thin muscles, right? There's kind of a substantial difference between these two types of athletes. This is a type 1. This is a type 1 athlete over here. This is a type 2 athlete over here. Very different looking. And so like that's what we're seeing, right? One is much larger than the other. One is much better at powerful contractions. Think about like how powerful a contraction you have to make to sprint super, super, super fast, right? You're really pressing really hard. So that's different compared to like creating a little minimal effort. If your duration is like greater than 2 minutes, especially if it's greater than 2 minutes, you're working on your type 1 muscle fibers. because you're relying primarily on your mitochondria for energy production. Remember, glycolysis lasts about 30 seconds. Glycolysis is really good for our type 2 fibers. So type 2 fibers, they are going to be divided into kind of two different components. There's sort of two different sides to type 2 fibers. You have got, type 2 means fast twitch muscle fibers, but you're going to have 2A and 2B or 2X. It was 2B when I learned it. Now they call it 2X. So muscle fibers that have a higher calcium level and more cellular volume. So type 2 fibers are larger in size, which means they have more cytoplasm and they have greater storage of calcium. So there's a greater storage of calcium ions, which we know creates our contractions. The increased cellular volume means there's more space to perform glycolysis, which is going to create even further lactic acid. But remember, lactic acid is a fuel, so that's a good thing. So these are larger in size. They don't have as much mitochondria compared to type 1 fibers because they're not producing sustained energy. They're creating short-term energy. But they do have more cellular volume so that they can do glycolysis. Because glycolysis is really, 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 really fast. Aerobic metabolism creates a lot of ATP, but it does it kind of slowly. It does it like in big chunks versus lactic acid versus cytoplasm, which is like creating very, tons of energy really, really rapidly, and then it exhausts its ability to do that. What's that phrase? The brightest candle burns half as long or something like that. The brightest candle is like glycolysis. So we're not going to see as much hemoglobin in athletes who primarily train for type 2. And then in their muscles, they're not going to have as much myoglobin. They don't need to deliver as much oxygen because they're not relying on oxygen for their energy production. They are also going to have more actin and myosin proteins within their muscles. Type 2 fibers have more of those contractile elements so that they can create those really strong, powerful contractions. These are used for your short-term, very powerful type contractions. Now, if we divide these into type 2A versus type 2B, the difference here is that type 2A are your fast twitch fibers, but they are oxidative glycolytic fibers, which means that they're kind of like type 1.5 in a way, right? Like they have a goodly amount of endurance, but they're still very much type 2 fibers. They're not as powerful as like a true type 2X fiber, which is like, I guess that's why I started using X instead of B, because it describes it's like extreme, Type 2X fibers are fast twitch glycolytic fibers. So you've got fast twitch oxidative fibers and fast twitch glycolytic fibers. Fast twitch oxidative fibers are kind of like halfway in between. They have a little bit of endurance, not as much endurance as like, a marathoner, but they have more endurance than like a sprinter. So they're kind of in the middle. When we think of like sports, we think of type 2A fibers. Like, when we think of powerlifting, here's the entire, oh, I don't know why I'm trying to pull this up. I'm going to get copyright struck again. Never mind. Power snatch. When we think about like a power lifter, their entire sport is this long. And he's done. Sport over, right? Versus like, sometimes you'll see like an NFL player, they'll be playing, for maybe like a 20, 25 second play. So generally we associate type 2A fibers with most of the sports that we're most familiar with: basketball, football, baseball, things like that. They're explosive, they're very strong, but they have a little bit of endurance versus your type 2X fibers, which are true, very explosive, and then they need a 500-year nap afterwards, right? True fast twitch fibers, extremely explosive. These are most associated with things like powerlifting, Olympic sprinters. And then maybe you are an athlete who's like a football player or something. And like, maybe you're like a running back. You're trying to run through a line of other very large humans, right? Well, maybe you're going to use those type 2X fibers to explode your way through the line. And then your job is not to keep running anymore. Your job is to go, great, I got it, first down. You set the ball down, you go off the sideline, you recover for a few minutes, and then they bring in the other guy. Like, so it's not that 2X fibers are not useful in sports. That's not the picture I'm trying to paint here. But generally, when you think about like basketball, for instance, right, they're playing a lot of minutes on the court. That's very much a type 2A fiber situation. And then maybe they use a type 2X every now and then to do like a really big jump, They're probably not dunking the ball 16, 17 times in a row. Probably doing it once, and then they're going back to like a little bit of recovery. Now here's what's kind of cool. You can modify your fiber types. The more endurance training you do, guess what? The more your body will develop blood vessels to the area, the more your body will develop more mitochondria. You will get better at endurance. Or maybe you decide to lift really heavy and your body will go, wow, we need more actin and myosin proteins. And so it will create more of that. The body is very adaptable, guys. You just have to apply the right type of stress, which is why we talked about everything we talked about today. With understanding this anatomy in a little bit of a deeper level, you can start to make sense of why we would give someone 12 repetitions versus giving somebody else 5 repetitions. Or maybe we put somebody right in the middle at 8. All of them are good. They just have different goals in mind. You wouldn't use a hammer to drill something into the wall, right? They're just different tools to be used. And that's about everything I got for you guys today. How are we feeling?
Great.
Love that. Paula, how you doing?
Whooped.
Oh, what's that, Lorianne?
Whooped.
Whooped. Yeah.
This week has been like, this whole thing has been like,
Yeah.
wow. It's good, but it's just fast.
Yeah. Exercise Phys is, Paula, just so you're aware, is our most technical class. This one in kinesiology, those two feel like a lot. But, trust me, we're going to get through it together.
No, it's good. It's really good.
All right, guys. I'll go ahead and kill the recording.